Okay, we're back. Last thing we're gonna do in immunology class. Isn't that something? Yeah. If you're watching this video, you made it. We all made it. Okay, here's the content from chapter 11 that we're gonna cover. And the chapter 11 content is about macrophages. And there's a lot more to chapter 11 than we're gonna cover. And you'll notice that we didn't even do a lot on neutrophils, on mast cells, basophils, yeah, and all the other cells that contribute in small ways. Oh my gosh, yeah, you could write a full textbook on this. Yeah, we covered a lot of it. We hit a lot of the high points, and immunology goes on and on, even far beyond what's in the textbook. But I think this will give you a good flavor. Anyway, fine, macrophages. Now, the thing you want to know about macrophages is that there are multiple kinds of macrophages, depending on how they get stimulated. And we don't fully understand all the reasons why macrophages are of different types. You can show that it's down to having certain receptors that let them respond to certain things, but exactly what are the environmental conditions that produce one version of a macrophage versus another, those are a little less clear. We do know some parts of it, so I'll tell you the parts we know, but we're pushing up against the gray areas on the periphery of what is known here. So, you remember Th1 cells and how I said their main job is to fire up macrophages with that old interferon gamma. That's still true. But there's a population of macrophages that will respond, and it's the big population, and it's the important population for like fighting active infection, and so we call those M1 macrophages. And so M1 macrophages are made and supported by Th1s. They'll respond to Th1 cells. That's a better way to say it. They will respond to signals from Th1 cells, that signal being interferon gamma. Yeah. Then they will go off and fight infection with all of the many, many tools that macrophages have, some of which we'll cover over here. Yeah. Then there are other macrophages that will see that interferon gamma and just let it pass by. So they will not respond to interferon gamma, and we call these M2 macrophages. And they will respond to other signals from Th2 cells. So you get Th2 cells helping out the M2 macrophages, and you get Th1 cells helping out the big old M1 macrophages, which are the heavies that you send in, basically. So, all right, a little bit more about macrophages. Let's go. So one of the things that T cells can do is that if a macrophage gets infected, and this happens a lot more than you would think, diseases like SARS-CoV-2 can infect some macrophages. Diseases like Ebola, strongly macrophage driven. HIV infects all the macrophages. Yeah. Not all of them, but a lot of them. It's actually pretty common with severe disease. But then you've also got things like mycobacterium can get in there. So you can get bacterial infections just living inside these things. And macrophages are important because they do a good job of eating things and presenting them. And unfortunately, because they're out there, dealing with all this nonsense, they end up getting picked off by all sorts of pathogens. And so we need a mechanism to clear those out. And so this is mostly the focus of at least the early stuff. Like, how do we tell macrophages that you're not looking so good? I think you maybe need to lie down and do a little apoptosis or something like that. Let's see. Let's learn more. Okay, there we go. Fine. So here is our M1 macrophage. When they get activated by interferon gamma, mostly, then they'll start to release a bunch of things. So they'll release nitric oxide. Now that can break down and become a free radical. They'll release reactive oxygen species. There's a class of things like peroxides, for example. Actually, one of the components in bleach, the active component in bleach, is called hypochlorous acid, And I believe macrophages can release that as well. So just literally bleach your bacteria to death from inside your blood. Yeah. Still don't drink bleach. That's not good. But yeah, your macrophages make a little bit of one component. Yeah. And bleach is a mix of some other stuff. Yeah, they're going to have TNF receptors, so they're going to respond to TNF alpha, which they can make themselves. And they're going to release a little IL-12. And IL-12 is, this is just the fanboys and girls calling out for more TH1s. Yeah, give us some more TH1 helper cells. Yeah, that's the idea. So they've got their MHC class 1 to show a little bit of what they're making. They got their MHC class 2 to show a little bit of what they've eaten. And they got some B7 molecules to give that little don't you quit on me push when a T cell does interact with them. So this will give a second signal. And then, some of these things would be sort of like third signals, signals that will further fire up our T cell, get it all stimulated. Okay, so just by way of showing you some of the things that macrophages can do and the way they interact with T cells. So let's go through the parts. Just make sure we got all the parts. So there's our normal interaction. You see this is an interaction with MHC class 2. Because it's got the two little legs on there. And so if our T helper cell releases interferon gamma, that's great. It's going to fire up a M1 macrophage, and the macrophage will get better at killing engulfed bacteria. So it'll eat bacteria and destroy them. But if the macrophage is infected itself, so if it didn't just eat the bacteria, if there are actually bacteria in it that are living and doing quite fine, thank you, then the T cell, helper T cell, will actually have release a little bit of Faz ligand. And so that's going to bind to Faz, and Faz is going to lead to apoptosis. Yeah. And LT beta is also going to induce apoptosis. So that's really good when there is a store of living bacteria inside the cell. And so it's a way to try and... this one will clean up our macrophages and or just get them to show you whatever it is they've been eating, because you don't want a secretive macrophage. Nobody wants that. This will actually kill the macrophage if it's too far gone. And the helper T cell has to figure out what to do. And it's complicated, of course, and we're not going to go into all of it. But yeah, figuring out whether to kill a cell or help a cell is, that's pretty fundamental helper T cell kind of stuff. Anyway, once the helper T cell has recognized something on a macrophage, it's going to release some IL-2, and the IL-2 will, of course, stimulate other cells to divide, other T cells, both helper and killer T cells. The rest of these are just other pathways where we can have macrophages starting to work. So IL-3 is going to basically, along with GM-CSF, those are going to cause monocytes. So monocytes are like baby macrophages, macrophage stem cells. They have not yet turned into like M1 or M2 macrophages. They're kind of like a TH0 in that way, but they have some other functions. So those are going to get stimulated with IL-3, and they're going to get start to divide. And so that way you can replenish your army if you're starting to run out of macrophages and monocytes. These can also release like LT alpha and TNF alpha, which will cause macrophages to cross not blood-brain barrier necessarily, but like blood tissue barrier. It'll cross endothelial membranes to go and invade, because the site of infection may not be in the bloodstream, and macrophages still have to deal with this. These are the foot soldiers of your immunological army, along with neutrophils. They do most of the work, and we give T cells all the credit. Yeah. When, these macrophages are out there day after day, busting their butts. So, I don't know. Show macrophages a little love. Yeah. Every now and again. They can also, TH1 cells can release CCL2, and that will call more macrophages, or rather more monocytes in, which will then get activated and become macrophages, and which can then eat up bacteria or virus, and yeah, whatever's making you sick, whatever's making you sad. Okay, so how's all this stuff work? Well, here we go. I think we got 6 slides left. Yeah. So, so the ideal is that you get your Th1 cells will bind to the macrophages that are infected and will completely clear out the bacteria if they have like the tuberculosis bacteria. Because those are sometimes going to be in little vesicles, which we can merge the lysosomes with those and get rid of them. But sometimes they get loose and they're just swimming around the cytoplasm, kind of like the way mitochondria do. And in that case, that cell is basically infected and there's no way it's going to come back. So the bad thing that can happen is if the T cell makes the wrong choice and just says, let's clear out those vesicles and everything's going to be fine, but the bacteria were already in the cytoplasm, then you got an issue where the macrophage is not going to be cured. And if the T cell does not, show it the old Faz ligand, then the macrophage is not going to die, and so you're going to have an infected macrophage that is not dead. And what you can get with mycobacteria, with tuberculosis, are these blobs of cells. They call them granulomas. I believe they show up even on x-rays potentially. And they're just balls with lots of little T cells banging into these infected cells that are they've got the mycobacterium just floating around in the cytoplasm. Some of these things will be fused cells that have more than one cell fused together, and there are a couple different ways that can happen. Most of them not good. And so you end up with essentially, just giant blobs of cells that are difficult to get rid of and are infected. And so, that can lead to a situation where you can't now clear a pathogen. So, that's why tuberculosis is still a problem, because it basically can exhaust, it can kind of get around the immune system. It exploits the weakness of Th1 cells, where they don't just kill everything, they try to help, and that's where mycobacterium gets them. Yeah. Okay. Here's an example of one of these granulomas. Apparently, I think the little dots are maybe the T cells. And these are all the infected cells in the center by the look of it. Fine. Now, Th2 cells make or interact with M2 macrophages, which is a smaller population. And the thing they mostly seem to do is we know for sure they're real good at getting rid of some kinds of parasitic worm infections. Yeah, so if you're not all hepped up on ivermectin, then you got your Th2 cells and your M2 macrophages to take care of that for you. So how does that work? Well, so here's our M2 macrophage interacting with our Th2 cell. The Th2s are going to make IL-13, and you remember IL-13. That was the thing that goes to these epithelial cells and will cause more epithelial cell turnover. So we'll just start, because you know how the gut works, just keeps pushing out cells from the bottom, and they ride up over the top, and then they fall off, and die. And you just constantly keep those moving. So you're going to replenish your gut lining. And also, some of the other things that the Th2s produce are going to drive up mucus production. And so it's going to be slipperier and more protected, like a little protective layer. IL-13 will also induce smooth muscle contraction, which, when it's in your intestines, that's going to be just taken, peristaltic waves, and so we're looking at probably diarrhea. So we're basically trying to flush out any sort of parasites, but there are potentially really bad health consequences of this. In general, when people are looking at regulating macrophages, they try to sort of bias away from M2s and toward M1s because people don't deal with that many worm parasite infections anymore. 100 years ago maybe, but yeah, not here and not now. So they're not as useful. They're not quite like an allergy level nuisance, but yeah, their place in the world is definitely dwindling. Yeah. All right. Th2 cells are going to recruit M2 macrophages. They're going to do this by putting out IL-4, their super secret message, and a little bit of that IL-13 will also go to the M2 macrophage. And then that will make a bunch of other products and will increase smooth muscle contraction even more. So these M2 macrophages are amplifying the diarrhea, basically. It's just what you want, right? Th2s are going to produce IL-5, which you remember is like a special love letter to the eosinophils. And eosinophils will then start to make proteins that will kill parasites, and we don't need to know the specific ones. But yeah, that's a way to do it. And with IL-9 and IL-3 from our Th2 cells, which are produced in lesser quantities, you'll drive mast cells to be activated, and mast cells are also pretty good against helminthic parasites. So like this would also be a pretty good response against like fungus, like those lung fungus, something like that. Eosinophils and mast cells. So they have a purpose for sure, and at one point they were probably totally essential to survival, but now is probably less essential than some of the other parts in clearing up the kinds of diseases we face on a routine basis in these modern times. Okay, 4 cells to go, or 4 slides to go, 4 cells to go. Okay, what about the old Th17s? Well, they don't do much with macrophages, actually. So Th17s, remember, are going to call in neutrophils. That's one of their big things. So they're going to release the old IL-17 and IL-22. That's going to cause those epithelial cells in the gut to make some antimicrobial peptides. So that's good. One of the things Th17s do is they're like a first line of defense in the gut. Because, remember, the gut lining is basically the outside of you. All that food stuff basically is not in your body until it's in your bloodstream, and it does not get there unless it gets past your gut mucosa, and it's not supposed to. Only like amino acids and sugars and stuff are supposed to get across that lining. So the IL-22 is going to drive turnover of these cells, so we're going to get more production there, and you'll see that with like a norovirus infection. You'll just sort of clear off all the cells on here, and the body will have to replace the entire intestinal lining. It just gets shed. Also, weirdly, with ivermectin overdoses, you get just shedding of all this intestinal lining, and then the body's got to play catch up and make a bunch of cells to replace all the ones that it lost. All right. The IL-17 is going to activate some other cells, stromal cells and myeloid precursor cells, and these are going to go on and increase the number of neutrophils. So we're going to drive neutrophil production because, yeah, just like Th1 cells basically drove monocyte production, which leads to macrophages because monocytes differentiate into macrophages that then become M1 or M2 or maybe M3 if we believe in M3. Yeah, and so IL-17s interact with neutrophils, and they also drive production of more neutrophils. Makes total sense now, doesn't it? Yeah. That will then also induce some of these stromal cells and epithelial cells to produce the cytokines, of which there are several, which will attract neutrophils. So again, that's a big thing that Th17s are going to do. And then, Th17s are going to produce CCL20 that's going to bring in more Th17s to drive an even bigger response. Unless something like a Treg comes in there and shuts them down, or unless you develop like a Th1 or a Th2 response that shuts them down, this is just the way it's going to go. Yeah. Till they see IL-4 or interferon gamma, they're just going to keep multiplying and bringing in more Th17s. So, That's the idea. All right, fine. 3 slides to go. Let's see. So one of the ways that this signaling works, both IL-12 and IL-23 have actually one of the same subunits. There's a P35 which is unique to IL-12, and there's a P40 which is the same. So it's actually 2 little bits of protein joined together that make an IL-12 molecule. That's just, so IL-12 and 23 do have those things in common, and so it's very efficient to produce both of these, for example. And they're going to bind to similar receptors. So the IL-12 receptor beta chain is actually going to be the same in both cases. It's the one that binds the P40 half. And the IL-12 receptor beta 2 chain is going to bind the P35 half, and the IL-23 receptor chain is going to bind the P19 half of IL-23. Do you need to know the P35, 40, 19, and 40? No, you do not. Do you need to know that IL-12 and IL-23 share a subunit in common? Yeah, that's a good thing to know. And share a subunit of their receptors in common. That's also a good thing to know. So anyway, that's going to lead to phosphorylation of various stat proteins, STAT4 and STAT3 in this case. And so IL-12 is going to produce Th1 cells, cause them to go down that metamorphic path, if you like, developmental path. And STAT3 induced by IL-23 is going to make us have Th17s, just like we covered in the T cell chapter, just like in chapter 9. Yeah. Okay, and that's just going to show you a little example here. So, if you got a RAG2 knockout mouse, now remember what RAG1 and RAG2 did? Yeah, these are the recombination things that make both antibodies and T cell receptors. So if you have a RAG2 knockout mouse, it's basically going to be deficient in production of both T cells and B cells. It'll be deficient in immunity in general. So if you take that mouse and you put in Th1 cells, so you can put in like an immune system in this mouse. And so this is just like how some of these immunology experiments work. You take a mouse that doesn't have an immune system, you add cells from your freezer that you've grown up all by your very own self, and so now you know exactly what these are. And you can make them, all reactive to the pathogen you're going to put in, for example. And then you can inject them with that pathogen. You can see what happens. So the idea is that Th1 cells can protect RAG2 deficient mice, which makes sense because RAG2 knocks out your ability to make T cell receptors, and Th1s are super important. Like CD8s are most important. They're number 1. Second most important are going to be Th1 cells. Yeah. For clearing out active infections of some sort. That works. And so they basically don't develop lesions at all. Whereas these IL-12B knockout mice, so they're missing part of IL-12. So they're missing part of the IL-12 receptor, and those have T cells that are differentiated, but they can't receive the IL-12 that would actually generate their own Th1 cells, and so they don't do as well. Don't remember this particular experiment. Just understand the gist of what's going on here. This is what immunological research is about. Yeah. So a little summary card for the end, because why not? So IL-12, remember, has one subunit in common with IL-23 and one subunit of the receptor in common. That's going to be an inducer of a Th1 response, which makes interferon gamma. And let's see, TL2 or TH2, sorry, TL2, TH2. TH2s are going to be driven by a bunch of things, including these, but they've left off like all the obvious ones, like IL-4, and they will produce IL-4 in addition to IL-5 and IL-13. So that's fine. That's what they're showing. And so these cytokines, if just these cytokines come in, even if the T cell doesn't make contact with an antigen presenting cell, the T cell can still release some of the cytokines that it would release if it were actually in contact. So it can make a partial response which is lacking certain things. So if we don't get IL-4, but we get TSLP and IL-33, then we're not going to make IL-4. We're just going to make a little IL-5 to fire up those, what was it, eosinophils, I think, and IL-13, fine. And same thing, if Th17 cells aren't getting the things that they need, they're just getting IL-23 and IL-1, then they're going to produce their IL-17 and their IL-22 just as if they were activated. So this is why IL-1 is pretty pro-inflammatory. It can set off Th17 cells to the full extent. They'll make all the important stuff that they would make if they actually were recognizing an antigen. And so an IL-1 response can relatively easily get out of hand. And too strong IL-1 responses are often associated with like fatal immunopathies, like immunological diseases. So there you go. I think that's the end. Oh my gosh, yeah, that's the end. So there you go. Immunology, man. There's a lot of it. I hope you got something out of this. It's complicated, but if you study it closely enough, I think everything would make sense if you only knew exactly which cells met which other cells and made which other markers in what concentration and which other cells received those markers. You can imagine a computer being able to predict very nicely exactly what would happen. But a lot of those events are down to chance. You release a little interferon gamma, you don't know if it's going to hit the interferon gamma receptor on the cell that you want it to hit. Yeah, in the part of the body where you want it to hit. And so, yeah, it's good, but it's a little haphazard. That's immunology for you. All right, thanks a lot. Thanks for sticking through the entire course and all these videos. Yeah, I'll see you later in this class or another.